The overall goal of this protocol is to provide a simple procedure to assess the relative abundance of histone post-translational modifications, which play essential roles in chromatin biology, such as gene expression regulation and chromosome condensation.
This method can help answer key questions in the chromatin biology field, such as which histone modifications change their relative abundance upon stimulation or treatment of given cells or tissue. The main advantage of this technique is that by using mass spectrometry-based proteomics, we can simultaneously quantify most of the known PTMs on histone proteins in a single analysis.
Histone modification analysis has numerous applications, including estimating epigenetic changes and chromatin aberration of individuals or model systems upon drug treatment or stress.
In addition to Simone, 2 other members of my laboratory will demonstrate the procedures of this protocol: Natarajan Bhanu, a research specialist, and Karch, a graduate student.
As shown in this workflow, this is a 10-step protocol that includes histone purification from cell cultures or tissues, histone derivatization, digestion and desalting, and mass spectrometry analysis using nano-flow liquid chromatography. Only selected procedures will be demonstrated in this video. To begin the procedure for isolating nuclei from intact cells, thaw on ice cells that were previously harvested and stored at -80 degrees Celsius, and prepare the nuclear isolation buffer, or NIB, as described in the protocol text. Remove 1/5 volume of NIB and add NP40 alternative to a final concentration of 0.2%. The remaining